Oh, no, it wasn't. I wonder why it's stopped
Okay,
recording.
now it's there. It wasn't a few
Okay.
minutes ago. Okay.
Huh, that's weird. Okie dokie, kiddos. So today is just a review. We are going to review two topics. First one is going to be cell cycle. So just want to know, what do you remember from the cell cycle? So there's 5 questions here. We're only going to keep them here for 1 more minute. Just type in the chat. How much of this do you remember? How much of this do you remember? Try to get your old noggin back into the groove of the cell cycle before we start. The record's, again, 4 out of 5. I don't expect you guys to get 5 out of 5. Maybe you can. It'd be champion if you did. Champion. Most people are getting about 2 to 3 out of 5. Let's see if we can get 5 out of 5. You've got less than a minute left. Come on, come on, come on, come on, come on. How much of this can you remember? How much of this can you remember? Come on, come on, come on. Hurry, hurry, hurry. Good job. Let's see, you're doing 2 or 3. Number 3 has 2 answers. Number 3 has 2 answers. There's 2 answers to number 3. So Luis, go ahead and give me your other. Which one is that for number 3? Hurry, 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 hurry. I'm going to wait for you to give me your updated answer, and then I'm going to take off. Hurry, hurry, hurry. I want to see what you get. Somebody else might beat Luis. Somebody else might beat Luis. Somebody else might beat Luis. Hurry up, Luis, hurry!
So I have a response from Carla.
You do?
Mm-hmm. Do you have Carla's? I can send
I
it.
do not. I do not. I do not. I do not. Hurry, 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 hurry. Give me the other. Give me your update, man. Oh, got you. Yeah, well, you were too ambiguous. I don't know when you say. Oh, I see from Carl. That's from Carl. That's from Carl.
Hmm?
1, 2, 4, 6. I don't know when you say one is asexual or one is sexual and the other is asexual. When you say one and the other, I don't know what that means. Okay, we're going to begin. It is time. So, good job. There were 2 of you guys that participated. So for participation, you get something at the end of class. Good job for participating on the Do Now. You get, maybe there's some secret points that you can get here today. So there's extra credit. And Luis and Carla, for jumping in there and giving it your best effort right off the bat, got something for you at the end of the class. I'll text you and tell you what that is. So there is a Do Now, which we just did. And now I'm going to go over something for review. Just going to try to review things really quick. And then you're going to have practice. You're going to have some practice. And if you get the practice today, you get the practice correct, then you are going to get 5 extra points on the exit ticket. Now, the exit ticket today is going to replace a lower grade on your exit ticket for October 30th. A lot of you guys have asked, can I redo exit ticket for cell cycle? Well, it's more than 3 weeks old, so no. But today you get an opportunity to drop that grade and replace it if you score higher on today's exit ticket. But you don't want to take it during class. You want to take it after class, because we're going to review 2 topics. We're going to give you some reminders about next week, and then Dr. Smith is going to use a quiz to review you guys over Punnett squares. So get ready, hang on, pay attention, take some notes, and you might be able to score some extra credit for the exit ticket, which will replace a lower grade. So first, mitosis versus meiosis. Let me go over some points here. So we're going to start out with mitosis with a parent cell that is diploid. You can see they're both, in this case, diploid cells. They have full sets of chromosomes. Mitosis only has 1 round of division that produces 2 daughter cells. Meiosis has 2 rounds of division that produces 4 haploid daughter cells. They're only 1 N, where the daughter cells for mitosis is going to be diploid or 2N. These are identical daughter cells. These are non-identical daughter cells. They are all going to be different. The daughter cells for meiosis are going to be haploid gametes, so it's like eggs and sperm, where daughter cells here are going to be.
Mr. Pub, I'm sorry, I accidentally hit mute.
Okay.
My
The in mitosis, I have daughter cells that are there. I have two daughter cells. Again, it's a little more basic of an image for it, but I go from a parent cell to 4 daughter cells in meiosis, parent cell to 2 daughter cells in mitosis. Mitosis are going to be identical diploids. These are non-identical haploids. So before we get started, play a short little 6-minute video for you guys, and then I'll review everything from this video. And again, pay really close attention to this, because you do get, this will replace a lower grade if you participate. And you do have to be present to win. What are we here, right? So let's see. And let me make sure the sound is going to work. Yes. I'm going to share sound on computer. Here we go.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, 1 came from mom and 1 came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make 2 new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form 2 cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with 2 identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells, gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Man, they do a good job. I really do enjoy their videos. They're really great for freshman biology. I hope that you guys see some of those that Dr. Smith has posted for you guys on the review. I do think it's important that you read, right? There's some things that are going to help you in a lot of ways that only reading can actually do for you. Okay, you should be able to see the PowerPoint again. I'm going to go over that information that she just went over, or at least the Amoeba sister went over. So mitosis versus meiosis. Just a quick review. Mitosis, there's going to be one round of division. You're only going to have one nuclear division. Meiosis is going to produce haploid cells because it's going to have 2 rounds of division. Interphase doesn't happen again in meiosis. Interphase happens before both of these. The products of mitosis and meiosis are pretty straightforward. You get 4 genetically different haploid cells. I know they're haploid because it's one N, it's just N, where in mitosis, they're going to be identical daughter cells, a result of somatic cells, body cells that are identical. They are both diploid. They are 2N. Going by the numbers, remember, diploid is a full set, haploid is a half set. And so we'll do a little practice. The symbol is 2N and N. So let's do a little practice. Haploid versus diploid. So if a human's diploid number is 46, our haploid number, since it's half, is 23. So if the diploid number in a horse is 64, what is the haploid number? Now you're going to get 10 minutes for practice, but only if you guys participate. So there are in this class right now, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 of you guys. I need to have 10 people take a stab at this first example to progress. And if you don't do it within a minute, I'm going to take a minute off the time that you guys get to practice. Good job, Carla. Good job, Carla. We need 10. We need 9 more. It takes longer than a minute. You guys will get 9 minutes instead of 10 minutes. 1 minute to take a guess. And you don't have to get it correct. You just have to take a chance at it. And you just drop it in the chat. Drop it in the chat. Good job, Chaparro. Thank you. I need 8 more people to participate. Xavion, good job. Right now, Xavion, Julian, and Xavion, excuse me, Carla, Xavion, and Julian are pulling for you guys. Good job, Xavion. And you're correct. All 3 of you guys are correct so far. Good job. Good job. We've got 3 so far. Push each other, push each other. If they're not participating, they're cutting time off of you. You're cutting time off of each other. Good job, Gio. That's 32. Excellent. So Gio, there it is. Gio, Jacqueline, Alejandro, 1, 2, 3, 4, 5, 6, 7, 8 people. Not quite enough. I'll give you one more chance. I'll give you one more chance. Let's see how this worked out. 100% of you guys that took a chance on it got it correct. So if a horse is a diploid 64, its haploid is only half of that. So how do I find that? I take 64 and divide it by 2, and 32, excuse me, divide it by 2 to get 32. That is going to be my haploid number, 32. Let's give you guys one last chance, not another chance, but a last chance at participating. So far, Gio, Xavion, Julian, Carla, you guys, Jackie, Alejandro, Cash, you guys all supported the class. Let's see if we can get at least 10 more. And you can look and see who they are. That would cut you out of your time. Still have 10 minutes. You'll get 9 minutes for practice. But you've got to handle the cat. So a cat diploid number is 19. No, excuse me. Haploid number is 19. What is this diploid number? So in this case, we use division. And we're going from diploid to haploid. Now we're going from haploid to diploid. What would we have to do if we use division this time? What would we do the second time? Give it a shot. Whoa, whoa, whoa. Good, quick, they're already doing it. Carla got it correct. Xavion, take another chance at it. Chaparro, you got it. But I got 3 participating. Got 1 minute. You want to, what would you do to go from, ooh, good job. We got 4. Got 4. Ooh, got it. Good job, Xavion. You got it correct. Yes, sir. Yes, sir. Yes, sir. Yes, sir. All you got to do is take a shot to support the class. 38 seconds left. No, excuse me, 28 seconds. You're going the wrong way there, Cash. You've got to get, a haploid number can be an odd number, but a diploid number is always, good job, Jackie. It's always going to be an even number because it's going to be 2 halves of something. All right, there was only 8 of you guys again. Sorry, so we'll have 9 minutes. You can blame the other people who didn't participate. So 38, so I had 38, that was the diploid number. In order to get that, I just take 19 times 2, gives me the diploid, right? So no big deal. If we just have to get double that diploid number to get the haploid. I mean, you can double the haploid number to get the diploid number. It's sure is confusing to say. Okay, so reasons why for mitosis. I have to replace cells that are damaged. Easy. No big deal, hurt myself, I'll let mitosis fill in those gaps. I need to grow. Obviously, if you go from a big or a small organism to a larger organism, you grow and develop, you have to replace, or not replace, but use more cells to grow. And so we grow in numbers of cells. Cells can't get too big. If they do, osmosis and diffusion stop. So they have to stay small. So the only way to grow is by reproducing the number of cells. Cells can't get larger. And if you're a unicellular organism, asexual reproduction. For meiosis, it's going to be for only sexual reproduction. 2 gametes, sperm and egg, make a zygote. We all started as a zygote. So that's sexual reproduction. No, yeah, exactly. One of you guys asked, so it's not asexual. No, you can't. Meiosis is going to be only for multicellular organisms. It does increase genetic diversity. There'll never be another one of you. You get crossing over, independent assortment, which Dr. Smith will, goes over whenever she does dihybrid. You independently assort the alleles, and then you never know which sperm is going to come in contact with which egg. So that's random whenever that mating occurs. Last thing, meiosis, body cells. That's going to be like your heart, your skin, your bones, those types of your parts of your body. Sex cells only come from gametes, only come from your, in this case, ovaries and testicles. So sperm and egg are only going to be generated in those organs, only in your ovaries and your testicles. So you have a Venn diagram. Let me show you where it is. You did have 10 minutes. But now we only have 9, puts a little extra pressure on you. So everybody should go to Schoology. Everybody should go to Schoology. And you guys are really lucky. You came to class, since you came to class, you get a chance at getting 5 extra points. And there's always been at least one group that's got 5 extra points today. And you'll get that on the exit ticket. How you get there is you go to class content, go to today's date. We're in December. Go to December, and then you're going to see 1, 2, 3 groups. I'm going to divide you guys into 3 groups. When you get there, you're going to open this document and you'll see you'll be in a different breakout room. So if you're in breakout room 1, you're in group 1, and you'll have this Venn diagram. And in this Venn diagram, you have some terms below. You have to put these terms in their correct location. So in this case, going from diploid to diploid, where does that go? Well, I don't know. It's either mitosis or meiosis. In order to get them in the correct location, you have to click on the image, double click, and you'll see there's a box here, a box here, and a box here. You just copy and paste the terms and put them in the correct location. The group that gets all of these terms correct in their correct location gets 5 extra points, and you're only going to have 9 minutes. Now, the groups that have done this today have been communicating while they've been in this. They've all opened the document, taken their mics on, and talked their way through it. Those groups have been earning 5 points for during this class for the exit ticket. Don't take the exit ticket until later. So I'm going to open you guys up to breakout rooms. And we can see if everybody's participating. It's pretty easy to actually see that. All right, you guys have got 9 minutes. Go. Hello, lonely person.
I didn't hear you.
Hello, lonely person.
Hello. I already have it open, so I'm just waiting on them to.
Oh yeah, I didn't put them. I didn't drop them in here because I didn't want to. Just. I wanted to give you a choice where you wanted to go.
Yeah, no, I'm good. I do see. I don't know if this will be confusing for them because it's labeled groups 1, 2, and 3, but their breakout rooms are 2, 3, and 4.
My God! Thank you for telling me that. I'm sorry. Holy crap! Oh,
I can go change their numbers real quick on their forms.
Yeah, do that. Well, shoot. Oh my God.
Or
yeah. Let me just go in there and just say something. It might be faster. I'm sorry. Hey guys. You're group 1. I'm sorry. You're group 1. You're not. I know you guys are looking for 4, but you're actually group 1. So use the first document. Use the first document. Yeah, group 1. Sorry. I'll give you the time back. Yeah, I just went to group 2 and 3, 2 and 3 were already in there. I went to group 4 and said, go to group 1. You're group 1, and then that's all.
and I just changed it as well. I changed it and I changed it on a document for group 4, group 1 to group 4.
Oh, okay. So it says group 4 now.
Yeah, it says group 4 on Schoology and on the document.
Okay. All right. Okay. Let me open that and see if they can see it. Hopefully they're in there.
They have not started. Let me refresh and there's something. Maybe I just don't see everything. Group 4.
You guys can see that she changed the document name for you, right?
No.
Well, I mean, you guys, let's see, this is. You guys should be able to see my screen, right? So yeah, you're in the right place.
The group 4?
Yeah, it's there. Yeah, you guys are in the right place. Xavion, Ulissa, and Mia. Yeah. All right, you guys go to work. Go to work. Go to work. Guys, you're not talking to each other. Y'all are not gonna make it if you don't talk. Let's see, group 2. Y'all haven't done anything yet? This is your chance to get extra credit, man. Y'all be hauling tail. You don't talk, you're not gonna make it in time. Now, you know, better start hustling, better start hustling. I'll leave. See, maybe you guys will talk to one another. Hey, Carla, were you getting any help in there?
Yeah, but we figured it out like literally last minute, so we didn't really do much.
Yes, ma'am. It took you guys, y'all had 10 minutes. What were you trying to figure out?
Because we couldn't put the words in the circles.
Why?
I don't know.
You just copy and paste.
No, because when I copied and pasted, it didn't paste onto the thing.
Hold
We
on, let
had
me
to
look.
like we had to pull out like this little box and then write in the box.
I'm doing it right now. Worked like a champ for me. It didn't, I mean, it was no problem. I don't know why. Are you on what kind of document, what kind of computer are you on?
I'm on the school computer.
Hmm.
But also Julian couldn't do it either, so.
Oh, that's weird. Well, Julian, you and Luis were the only people, and is Luis still here? Luis is not here anymore. Oh man, you hung on. So you came in and you were the first person, one of the only two people that took a chance at the do now. So since you did that, you get 5 extra points for taking a shot at the do now. So you tried everything that we did today, Jackie. So you get 5 extra points on the exit ticket today as well. All right, guys. Nobody got that done. Nobody got that done in time. Again, but I would go and I would order it myself before I even took a shot at trying to take the exit ticket today. I turn it over to Dr. Smith, and she's going to review Punnett Squares with you guys.
All right, awesome. So let me go ahead. I'm just going to share this PowerPoint really fast with you guys to make sure we're all on the same page, and then we'll jump into the practice. You should be able to see the screen now, and this is our PowerPoint from before, so you can go back and look at it. We are looking at dihybrids and monohybrid crosses today, and we're going to do some practice with that. Remember, the dihybrid involves the 2 traits, and monohybrids involve 1 trait that we are solving for the potential probability of the outcome in the offspring as far as the trait is concerned. With that being said, remember you do need to identify the genotype of the parents first. And from there, if you are doing the standard way with the 16 squares, then you're going to have to go ahead and solve for the parental gametes using full method. And then you set up everything with the 16 boxes. So with 1 parent on top, 1 on the bottom, and then you fill everything in, which will look similar to this, and then identify whatever phenotype ratio is asking for. So that's the one way. The other way is using the rule of multiplication. And for that one, you're still solving for 2 different traits, but you're using 2 individual monohybrid crosses and then setting them up, each trait having its own cross. And then you're solving and multiplying them together to still get the same outcome. So those are the 2 ways that you can solve for the information. This is a faster way, especially if you're doing an assessment. So I'll stop sharing this, and then we'll go ahead and look at the practice for today. Make sure you have a pen and paper because you will need that to solve for some of the questions that are being asked. All right, so go ahead. You should see the joinmyquiz.com and then the code. So go ahead and log into that. And the way that this will work, we will have a 1st, 2nd, and 3rd place winner. And that just means that you not only answered the questions correctly, but you also did it in a short amount of time. So it was accurate and efficient. And let me copy this, and I'll drop this link in here as well. So if it's easier for you to use the link, you can do that from the chat box. All right, so there's the link. And so 1st place will have an opportunity for 10 bonus points, 2nd and 3rd place for 5 bonus points each. And I'm going to go ahead and set the timer for 45 seconds. Please go ahead and sign in, and we'll get started. Take 35 more seconds if you have not signed in. So waiting on 2 people. I think we lost somebody.
Don't know.
Thought we had 11, but maybe I'm remembering wrong.
I thought we had 9. I think 11 including us.
Yeah, and now we're down to 10 total. If you haven't signed in, waiting on 2 people, you have about 5 seconds so we can get started. So again, make sure you have pen and paper. All right, let's go ahead and start. Looking for wrinkled and green seeds. Waiting on 1 person, be mindful of your time. Make sure you identify what's the dominant trait and recessive trait for each of the traits specified. So we're looking for wrinkle versus smooth, green versus yellow, and then from there you just identify the offsprings. You need to identify your genotypes and then set them up in a Punnett square. Looking for the genotype for the offspring. So this one, you're going to use this one to answer for this question and the next question. So make sure you jot down the genotypes for the parents. Go ahead and set up your 2 monohybrid crosses or 1 large dihybrid cross and solve, because you're going to need to use this for the next one. Make sure you have the information you need here, because you'll need it from the same info here to answer the next question. So wait 5 seconds. All right, so that was the last one. Let's see.
Who won? And they had a high probability of winning in this class. 7 kids participating. 3 are going to win, just almost half that extra credit. Ooh! I would have definitely joined this one. Winner, winner, chicken dinner. Jackie.
Jacqueline. Who was the first? No, Jacqueline was first place. Who was second? Julian. Okay. Congratulations, you guys, on that. And Jacqueline. So writing that down, make sure. All right. Make sure you go back. Let me go ahead and share. We'll stop sharing here. And just go back to the other screen really fast. Remember on this, you guys, because a lot of you all missed this one. Just real quick, when you are looking at, where did it go? When you're looking at this and it asks you about the ratio, like how many are, let's say here, if it asks you how many are brown fur and straight ears, if it doesn't give you a key, you have to set up and identify your genotype. So you have to identify like what's the dominant, what's the recessive for each one of those. And once you do that, then you answer for whatever the question is asking. So if it's asking for brown fur and straight ears like here, then brown fur, we know it has to have a capital B if that's the letter that you're using for that example. And it's going to either be a capital B for dominant or a capital B, lowercase b for dominant. And it also asks for straight ears, and straight ears also would be, is a dominant trait. So it would be capital E, or capital E, lowercase e. So just depending on what's being asked, and then you go through your chart and you find that particular combination for traits. So brown fur and straight ears. And then you just count them up. And the total number here is 16. So it's out of 16. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12 that fit this particular description for brown fur and straight ears. So it would be 12 out of 16 for that. So that's how you're getting to your answer choice there. If they've given it to you, or if you're having to set up yourself, you set up the genotype for both parents, and then you basically go through the solve. You can do the long way like this, or you can do the short way of multiplication where you set up two individual Punnett squares, monohybrid squares, and then you still solve. Once you get the information, the only difference here is you just, if it's brown fur, it's 3 out of 4, 1, 2, 3 are dominant here. And straight is going to be straight ears, 1, 2, 3, 4, all 4 of those. So it would be 4 out of 4 and 3 out of 4. You multiply those together in order to get brown fur and straight ears. So it's just two different ways, and that video is still available for you as well. If you want to go back and look at that video again, or the lesson from that day. And let me go ahead and share in Schoology where all of that information is for you so that you can do your review. So you should be able to see my Schoology page at this point. Make sure you're going back to class content. Once you go to class content, then you'll see here where it says Q2 benchmark review resources. And these are the resources for the benchmark for B-Day. You guys take the test on next Monday, the 14th. We're covering cell cycle, DNA and RNA, transcription and translation, and then genetic outcomes. For each one of those, if you open up this folder, there's a subcategory folder for those particular lesson topics. And in each one of those, you will see video summaries of that information for that particular section. There's a study guide there with some practice in there as well, and vocabulary. And I'm actually going to go back and add just some background reading. The background reading is in the individual folders for the lesson day, but I'll just go ahead and drop that in here too if you want to go back and look at that. And you can do that for each one of the topics, and you can review as detailed or as general as needed, depending on what you need to do independently. Any questions for any of this? All right. So if there are no questions there, then Mr. Pope, I think you.
Yep. So yeah, there's, if you look at the bottom of her page, there's the cell retake. This is for quarter 2. There was a cell test in quarter 1. Some of you guys have asked to retake that. We set a date after class on Tuesday for you guys to retake that, but nobody came. If you would like to take it now, you still can, but not now, like at this moment. What I'll do is under Khan Academy, you guys know that I have you guys signed up as a class. You signed yourself into a class. I'm going to post some review resources. If you want to take this, do those review resources and say, I finished those review resources and I would like to take that test. You have until Sunday evening to say, hey, I finished those review resources. I'm not going to post those review resources until about 2 o'clock today, about 2 hours from now. But if you do them and you say, I want to take that, then, you know, send me a remind message on Remind, and I'll release that test to you to take. Okay, that was all I had. But in the meantime, I hope that you guys have a fantastic day. Man, that's the last class. We will make sure that you are there in class on Monday, because you only get one shot at taking the final exam. You don't want to miss that. You miss that, you're really in trouble. So make sure that you're there on time on Monday, not late. You guys be good.
You guys have a good rest of your day and a great weekend. Thank you.